So Richard he trained in math, and then he was at the LMB. He tells me he went off to do some neuroscience before John Solston got him back to the LMB, and then Richard moved with John to the Sanger Centre in about 1992 when it was formed. So he's a founding member of the Wellcome Trust Sanger Institute, and has been there since the very early days of the Human Genome Project. And I was delighted to look up the PubMed and see that we've actually co-published in 14 different papers. So some early human genome papers, the RAT project, but then in HapMap, but then the 1,000 Genomes project, which Richard really initiated and led and was the prime mover. So today he's going to talk about the extensions of those large-scale sequencing projects, and I believe his title is more esoteric. It's all things about sequencing. So thank you for coming, Richard.
Okay. Well, thank you very much, Richard, and thank you to the organizers for inviting me and for the meeting. I have really been enjoying the meeting, and I hope I have something useful to say to you. So there was a different title in my abstract. Of course, I was asked to give that some months ago, and I gave a sort of generic title. And then I've ended up with still a rather generic title. But I've been thinking, where we're going with all this sequencing. And in some sense, that's the topic of the meeting. And I know a lot of it is focused on how that's getting translated out into applications beyond research in clinical applications in medicine. And I'm going to rather think about where are we going with respect to what we can do next with it technically and in terms of understanding of human genetics. But I think that will feed back very rapidly into the medical and additional applications. Okay. So there's a sort of standard picture that one has of the rate of increase in sequencing and decrease in cost, so that the first human genome took a long time and was very expensive. In 2009, there were about 200 human genomes sequenced at the start of the early 1,000 Genomes project, about 50,000 pounds a genome. 2012, thousands available sequenced for research, beginning to be used in all these other areas, costs around $5,000. Maybe next year, $1,000. People were right saying it's sort of flattened off, but there's some hopeful things, I think, right, not even on the horizon, but sort of within view. And I'm pretty sure there's enough of them that I'm sure there'll be a competition to really get down by another factor of 5, if not an order of magnitude. And we're going to be using them for research, clinical use, of course, and I think increasingly personal interest, and some of that people got into in the last ethics discussion. And this means that by 2020, we should expect there'll be millions of genomes sequenced. And what will be the consequences of that? What are the opportunities and consequences, I think? So the growth, much faster than Moore's law, stretches us all. It has done and will continue to do so. So I want to talk about three things, three ways to use more data. So the first is really to increase the power of association studies. And they've been a powerful way that modern genomics has got at gene function in humans. And there are thousands of associations which really, whether or not they explain all the variance in phenotypic diversity amongst people, they are, I think, solid results that tie the variation at a particular genetic locus to some phenotype in all sorts of interesting ways, and people, they've been a major step forwards. There's been a discussion about how the association era, which in a way started with genotyping and with HapMap that Richard referred to, how that's really going to move in the era of sequencing. I'll talk a bit about what I see happening in the near future in that area. Then I want to talk about, as we get up towards sequencing a million people or millions of people, are we going to start to see saturation in human mutations, and what will that mean? And I think it's worth thinking about that. I think that will change our kind of view. At the moment, people think you identify, people draw these pictures of genes, and you find a few variants scattered around, and it's sort of a bit of like a shotgun sort of activity where you may hit or miss particular residues. And that's really a consequence of small samples. And we're going to get towards saturation sooner than I think people think. And I think that will actually really lead to a change in viewpoint. And then that's got to do with just finding single mutations by themselves, which are normally heterozygous. But anyone who's worked in genetics, particularly model organism genetics, knows that really to understand the consequences of mutation, functional consequences, often ones look at the homozygote. That seems kind of inaccessible, but actually it's not, because of the substantial amount of consanguinity in the world. And how can one leverage that to really get at human gene function on a systematic basis in the same way that people had thought about in the past in model systems? So I'm going to talk a little bit about the 1,000 Genomes Project. It's just a sort of lead-in, and maybe there's my perspective on that has some things to say for people. It was a consortium that was founded now quite a long time ago, actually in this room, really proposed. Pilot projects were published 3 years ago. About a year ago, the phase 1 with over 1,000 samples. We have the complete data set now on 2,500 samples, and are working hard to get that all finished and completed and published next year. And the aims were to find essentially all the variation down with some, down to some allele frequency in the population, notionally at 1%, which was the kind of classical concept of polymorphism, and do a better, more thorough job finding rarer variants, particularly in coding regions. To genotype them and place them on haplotype backgrounds, which really means to identify not just that the variant existed, but to actually give a version of the sequence for each of those individuals, which we could then use in all sorts of other analyses, and to find all the other sorts of variation present. And it's been a driver for both data production and methods that are used now by all these other projects. So I can kind of zip quickly through this. These are the HapMap populations that formed the pilot phase. Phase 1 that was published last year involved populations from 4 different continents, I guess. We moved into a model of a set of populations with 100 individuals per population, a mixture of whole genome shotgun at low coverage and deep exome sequencing. And the final version has data from 26 populations, 2,500 samples. There's actually 6 populations in the Americas contributing about 500 samples between them. As I said, data collection is complete. And I think for the first time in this sort of integrated fashion, we've brought in variation in South Asia that's obviously got a huge population and a rich source of diversity and involved in medical studies, as well as far from complete, but a broader representation of variation in Africa. This is by no means yet a complete picture of variation across the world, but it's a useful position. So as we've done that, we've found more and more variation, of course, as we've sequenced more individuals. The final, it looks like we're going to get about 80 million SNPs in the phase 3 final version. The accuracy has been improving. The number of false positives has been dropping. It's a very rich data set with all sorts of structure you can see here, and these long bars are deletions that are shared by some individuals, and there's, you see the African populations have more variation than some of the other populations. A lot of structure, genotypes. There's now towards 100 terabases of raw data involved in this project. And there's one more 1,000 genome slide, just to sort of show you, as we expect, as you go to lower and lower frequencies. So this is in fact from the pilot. There are more and more variants. This is a plot we've seen many times. We saw very nicely yesterday. Because of the low coverage sequencing, we haven't observed every variant at the low frequencies. But then, of course, because of sampling, we also don't observe every variant. Things that are just a singleton in the sample that we have, from a population point of view, there'll be other things that are present in the population that we wouldn't have sampled. So if we think of this as a population sampling strategy, the low coverage strategy is appropriate. It's the most efficient design for telling us about the variation in the population, for spreading our sequencing across samples from the population. And that's a very different situation from a medical situation where you have an individual and you really want to know what their sequence is. But even there, I mean, there was some discussion yesterday that one should, Debbie was saying you should double and increase the amount of sequence you get per person. And of course, you can always tell more about that one person. But sequencing, resource allocation isn't for free. It's not infinite, however one might like to think, for each patient. And I think even in the medical setting, it makes sense to think about tiered approaches and most efficiently using the sequencing technologies across the range of people for whom it would be appropriate to use them for diagnosis, and the most effective ways to do that. So this picture on the right comes from the phase 1 data, and it's looking at variants found in another project, the UK 10K project, where we know the allele frequency in that project, and that's in 5,000 across 2,500 individuals, 5,000 haplotypes. And you can see we're getting 96% of the variants at 1% frequency, which is what our goal was. We're also finding half of the variants at 0.1% frequency. So that's actually an important message about the 1,000 Genomes Project. It isn't just finding things down to 1% frequency. It finds in fact, the vast majority of the variation found is at lower frequencies. And it's doing quite a good job of representing even this kind of lower frequency range. Of course, there are some singletons, but you're getting a very small fraction of the extremely rare, ultra-rare variants. Okay. So that moves on to, as people develop these technologies and we started to know that we could access this low frequency data, the question came, can we use those sorts of approaches, sequencing approaches, to get at the functional variation that's present in the genome at lower frequencies? So here again on a log plot, on the x-axis we have allele frequency. This is a sort of standard type of picture that you've seen many times, where you're seeing rare variants with large effect that were found by linkage, common variants with typically small effect that were found by association studies. There's not much that has been found that's common and has very large effects. That's particularly on disease. And that's not so surprising. You'd imagine that selection would remove anything deleterious in this region. It's going to always be very hard to find functional effects of very rare variants which have almost no effect. There's a basic statistical problem. I mean, we may get there. We'll push things down in this direction as we go to larger and larger samples. But there's a big hole in the middle here, and there was a feeling that this is exactly the range that we can access by sequencing, and that there should well be or may well be material in here that is important, both in terms of the contribution to disease, but also into our understanding of the genes involved. Because genes here are a bit haphazard. It's the ones that happen to have common variants that have effects. And we've known for some time that there are some things here because of candidate approaches. So PCSK9, which we'll come back to right at the end, is a gene where there are variants with a fraction of a percent frequency that affect LDL, cholesterol, blood levels, with quite, with moderate effect levels, so that overall they sit in this domain. So a number of sequencing studies were set up a few years ago, one of which I was strongly involved with, the UK 10K project, to sequence a few thousand individuals, the same sort of order of magnitude as the 1,000 Genomes project, which was becoming feasible for one big grant, to ask questions about what you find in this territory. There have also been exome-based studies, but they've been a mixture of exome and whole genome-based studies. So those are sort of coming. You probably have, many of you will have heard talks about those. They've been sort of, I guess, the general consensus is somewhat underwhelming. I think there are some messages and some stories that have come out. There are some variants with low allele frequencies with moderate effect sizes. There's some up here which were missed because they weren't well tagged, between 1 and 5 or 1 and 10% frequency by existing things. But I think actually the benefit of these projects is going to turn out to be not genetically in terms of what they find themselves, but of the resource they build, they've created in terms of genetic infrastructure. And I want to talk a little bit about this project, the Haplotype Consortium, that's been led really by Jonathan Marchini from Oxford, involving a whole lot of collaborators from different groups who were involved in these large-scale sequencing studies. And the aim of this project is essentially to combine all the information from these various projects so as to give us a reference resource that we can use in combination with pre-existing GWAS data or with GWAS array data to reach out into this region of the space. And I think what we're going to find is that the message that we find is that one needs very large sample sizes in this to find things in this region. We actually have those sample sizes in big studies, epidemiological and case control studies, and many of those samples have been array genotyped. Rather than sequence them all, we're going to be able to use reference panels to impute into those, and we're going to be able to impute in this territory, I think. So this is a brief sort of introduction from Jonathan to imputation. You can think of having a set of genome sequences from a reference panel, a set of genotypes that you've obtained from your study participants, probably tens of thousands of them. You can match, you can identify which bit of this genotype belongs to which one of the reference sequences, and copy down the missing data to fill in missing data, and so impute complete sequences for these individuals. And these strategies have been used for several years, and they're used to help increase power, combine data across studies, and increase resolution. And sometimes, here's an example where the black things are the genotype sites and the gray are the imputed ones, and you get much more information about what's involved. So what's the history of this? Well, they started out imputation with HapMap 2, 2 million variants. The 1,000 genomes pilot gave 15 million variants. Phase 1, about 40 million. The 1,000 genomes final will be 80 million, but at the same time there are studies on similar numbers of individuals, UK 10K, Go T2D, Go NL. Altogether here there's 15 million individuals and very large numbers of additional sites. So the goal here is to combine together all those different samples. We can, actually these are all currently sequenced samples. There's about 35,000 samples present around the world across different studies. Each one of these is at best marginally powered, but between them, this is an order of magnitude bigger than any previous data set. And how can we combine those data together, and what will that enable us to do? Well, there are technical issues involved in that, but I'm going to skip those here, really. We have a pilot project on chromosome 20. It's about 50th, 2% of the genome. 13,500 samples, 1.5 million variants. That would scale up to about 75 million variants across the genome. And there's quite a lot of missing data, and that's not surprising. It's because if you find something in one of these studies, it's looking at a lot of individuals, you may not find it in another study. Or the techniques used might have decided to exclude that site in another study because they weren't so clear about it. But most of that missing data is rare, and we can fill it in by imputation quite accurately, one way or another. So this is the take-home from that at the current state, and it's a work in progress. This is the line that we get from using the 1,000 genomes phase 1, and here again we have allele frequency on the x-axis and a logarithmic scale, and this is the R squared, the imputation power. So at 5% R squared, we can 5% allele frequency we can impute with 90% R squared. With 1%, we can impute at about 50 or 0.5%, about 50% R squared, 0.5, which is kind of useful but not really ideal. So with this pilot data, we're already pushing ourselves up to be 80% R squared at 1% frequency, and it's quite flat here, so we're getting out to the fractions of a percent scale, and that's looking quite promising. These for imputation in this area, which previously we thought would be accessible by sequencing and accessed by sequencing. If we simulate, then we think we ought to, with this data and with the type of data we're going to be getting, we should be getting at this level up here at 85% R-squared and really being able to impute out to 0.1% for all the variants in this range, which are really the range that we were thinking to get at by sequencing. So I think that's promising, looking forwards for using large haplotype reference sets in combination with very large genotyping data. And for example, the British Biobank of 500,000 people is all getting genotyped now. That's in genome-wide. So we'll be able to do GWAS studies on 500,000 people, association studies imputed into tens of thousands of individuals. And Tim Hubbard talked about the Genomics England project. That will give us 100,000 high accuracy reference haplotypes if they allow us to use them for those purposes. And we'll have to see whether we can do it inside their firewall place and recover back our inferred data. But there are some interesting ethics questions there, essentially about anonymization and not transferring individual data out. Because the whole aim here is not to say something about the reference, but to say something about the samples that you're putting in. Okay, so that's one direction I think we're going to go with a lot of sequence. So what about going beyond, down to rarer things? Most of what we've heard about have been really rare mutations. Can we see mutations? Yeah. And there's a feeling at the moment, we can find these mutations all over the place. Every time we look, we find new mutations. But everybody's still thinking in terms of taking your individual case, subject, and finding the mutations in that individual. And I'd like to put you in mind that I moved into biology in the 1980s, and that was before we had any genome sequences. People had no idea how many genes there were. But you would do a study, and you'd know that there was you'd find a mutation, you'd think there was a gene for something, and you'd know it was in a sort of sea of genes, and you'd fish that out. And every single study did its own extraction of the individual, of the genes of interest to it, just as now we're finding the variants of interest to ourselves in each individual. And we think of it independently going each time and getting new sites. Of course, we all know this fact. We know that with a per generation mutation rate of about a little more than 10 to the minus 8, a world population of 7 billion, there are about 100 de novo mutations per site across the genome around the world. And that's kind of a known fact that people think, well, that's inaccessible. Nobody's ever going to get the sequences of all the people in the world. And that's probably true for quite a long time. That's not, we don't really need to think about that. But it is relevant. I mean, that means mutations with strong dominant phenotypes select themselves. We know that. And we have seen that, we see the same mutation again. And Matt showed the examples of this yesterday within Britain by chance. All of you probably have seen this happening. So you really are accessing that whole population when you're working with a strong phenotype to pull something out for you, or some fraction of it, maybe a tenth of it in the good fraction. So what about inherited non-lethal mutations? Well, they accumulate over generations. We're not looking at at lethal or very dominant mutations that affect fertility. And there's been this population expansion in the last 10,000 years, about 300 generations. And that means that you can accumulate separately mutations over hundreds of generations. And if you can accumulate mutations over hundreds of generations, then maybe you only need about 1 million people to start seeing 10 to the 8. That would be 100 million sort of generation people, samples, and at 10 to the minus 8 per generation. That would be about 1 per base. We might begin to approach seeing a mutation every base. Matt said yesterday people were shocked when he said he saw a mutation every 20 bases. But I think I'm going to argue that we can get ourselves into a position where we'll see a mutation every base. So if we take a population model that is fit by some of the studies that people have looked at of about, these are kind of population genetic parameters. They're not the world population size within a functioning population. Modern population of about 10 million, decreasing going backwards to 10,000 about 10,000 years ago, and then a bottleneck, an out of Africa bottleneck. If you took a single sample mutation, say in Britain, say you sequenced 1 million people from Britain, you'd actually get a mutation on average about every 2 bases. But if you take 10 samples of 100,000 from different populations around the world, then you'd start to saturate. You'd get approach 2 mutations a base. And so by picking subsamples, and it's quite interesting that the way the world population is structured actually allows us to get at this in reasonable time. So this is a figure to show that. This is the generations going backwards in time. That's the out of Africa bottleneck about 2,000 generations ago. A couple of thousand individuals. This is the current, this is in terms of number of chromosomes, so going backwards. What you see is if you have 1,000 people, you're getting about, those people don't really share a common ancestor for thousands of years. That's about where we are with the 1,000 Genomes Project. We're looking at old mutations that are shared between individuals that were early on in the out of Africa event or predate that, or are private to individuals. But as you increase the number of samples that you get, you're sort of looking more recently. So you're looking back only about 180 generations per sample with 10,000 people. With 100,000 people, you're looking back about 100 generations per sample. And then you begin to saturate. You're not really gaining that much more because this is already too close to the population sample. So it turns out that you sort of do well to have about 100,000 people you pick, say, from Britain. You don't want 100,000 also white Americans because they're pretty much British if you go back not very long, or there's too much overlap. But you could pick populations from around the world, from Europe, the Middle East, South Asia, Native America, multiple African populations. And we could start building up a sample where we actually we're observing mutations at every base. So should we think about a Million Genomes Project? In some ways, I'm surprised nobody has done that. When we started the 1,000 Genomes Project, fewer than 10 genomes were available. So it was a factor of 100 off what we could do at the time. Well, we have more than 10,000. Sorry, I left out a 0. We have more than 10,000 genomes now. We're only a factor of 100 away right now. And people are talking about 100,000 clinical genomes. It's really quite reasonable to think certainly within 10 years, within 5 years, there'll be 1 million genomes. So that's one. I think people should think about this. Obviously, they're going to be driven by all sorts of factors, clinical factors. I'm not looking for another project. So it's not sort of something for myself, but I think the mixture, the combination, we should be thinking about alongside clinical applications, how that data, what that data could be used for and how it would be used. And also, when people started talking about matching variants, we should start to realize almost anything you discover in your sample will have been seen somewhere else, by somebody else. That's a relevant way to think. So that's, those are almost all going to be heterozygous if we take standard outbred humans. What about homozygotes? So we're not going to saturate in the world for homozygous mutations if we're thinking about people who are outbred in a big population. But consanguineous marriages, such as first cousin marriages, of course, as we all know, and we've heard about them, they lead to autozygosity, to homozygosity of shared through descent. A 16th of the genome is homozygous in the offspring in a sort of clean model. It depends upon the population history of the individuals concerned. And somewhat to my surprise, it's estimated that about 10% of all marriages worldwide are first cousin marriages. That number comes from Wikipedia, but there are references there and from other people that I've talked to. So some of you may be aware of this. I think others probably not. The level approaches 50% in some areas of the world, and it's also not something, in the Western world. In Europe, it was completely standard to marry your cousin. Only a couple of hundred years ago, Darwin married his cousin. It was a sort of double, like we heard about today, a double cousin marriage. Their siblings married each other. Queen Victoria married her cousin. And people in villages, commoners married their cousins at all levels of society. It was not an aristocratic thing. So anyway, it's clearly varies culturally, but is common. If we think of 10% of 7 billion, 700 million consanguineous individuals, a 16th of 700 million is 50 million homozygous chromosomes per site. So if you get something that isn't a lethal mutation, either or dominant lethal or recessive lethal, which of course would be not observed, probably, since there are several generations of accumulation before the shared ancestor, you're going to observe every site in the world. There will be, in fact, every site homozygous as well. So that's a sort of statement of what about the world. Are they accessible? Well, they're not accessible. You'd have to sequence all consanguineous individuals in the world really to start to saturate the per base homozygotes. But we can ask about the per gene. We can ask, and per gene there are many different ways of knocking out a gene. So we could ask about loss of function homozygous mutations in individuals. And of course the number of loss of function mutations, possible loss of function mutations per gene is very variable. There are small genes or very large ones. The number of splice sites varies. We know that indels are important, but they're low frequency. But there's something like of the order of 100 on average possible loss of function mutations per gene, maybe 50 to 100. So sort of at that scale, maybe with some history, maybe something in the tens of thousands or hundreds of thousands of individuals, we could start to find all homozygous loss of function variants in all genes in humans. So sort of thinking in that direction, there's a new project which is led by Richard Trembath and David Van Heel from Queen Mary, London, and the British Population Autozygosity Gene Function Study. The aim is to recruit subjects with self-stated parental relatedness, ideally first cousin relatedness, from populations with high levels of consanguinity. Particularly this is focused in the East End of London, where Queen Mary University of London is based. There are very large populations, hundreds of thousands of Bangladeshi and Pakistani immigrant and first and second generation populations with high consanguinity levels. Sequence the exomes, identify homozygous rare loss of function variants and others, and then they're specifically recruited under consent for recall based on genotype. So you can target genotyping at particular classes of mutation that can be clinically relevant to those individuals and phenotypically of interest with respect to the functions of the genes. And the phase 1 of this has been funded for 15,000 subjects by the Wellcome Trust. So we've heard about medical implications, but of course human gene knockouts are highly informative. They help us understand gene function, link genes to phenotypes. This is why people historically have made models of knockouts. But there are all sorts of issues in that of whether the model is a good model or not. Of course, this doesn't replace models. In animals, you can't do, there are all sorts of things you can't do in humans. And typically, you'll have small numbers of individuals. But already, human gene knockouts have proved important for all sorts of studies. There are sort of famous stories with PCSK9, the gene I told you about before. There are loss of function mutations. There's an individual, I think just one individual was identified whose loss of function is a compound heterozygote for loss of function mutations, has extremely low LDL levels. But the important thing is that he's otherwise a healthy person, and immediately tells you that this is a good drug target. If you could knock down completely the function of that gene in a targeted specific way, then you can lower LDL levels, and also there's no immediate harm comparable to a phase 1 drug trial. CCR5 is another example where there are homozygote CCR5 deletions. Gives you immunity to HIV. So we have some pilot data for this project from 1,100 exomes. If we look at the distribution of numbers of homozygous segments in red from this sample, in blue from the UK 1,000 genome samples, you can see there really aren't any in the UK 1,000 genomes GBR samples. There are here. There isn't a neat bump at exactly a 16th, which you might get. That's because there's a complex distribution of different levels of parental relatedness in the population. But there are clear examples of long homozygous stretches, as in this individual here. So here we've got a region where there are only homozygous variants and no heterozygous variants of 50 megabases, and another one on the same chromosome of 30 megabases or so. So in these we find 213 high confidence loss of function rare homozygotes in 198 genes. It's just in the pilot. And we see, as we might expect, that the higher the inbreeding coefficient or the homozygosity, the degree of runs of homozygosity, the larger the number of loss of function, rare loss of function homozygotes. If we look at the set that we found, these are all the high confidence loss of function variants. Many of them are het still, because it's still only a small fraction that are homozygosed. You have to be careful, as we heard earlier from Les, that you can't assume always that a stop codon. If you find a stop codon, it could be at the very end, and that could not really change the function of the protein, or it could be at the very start, and there could be an alternate methionine, or it could be not in a constitutive exon, and there are all sorts of other issues. So Daniel MacArthur has looked at this extensively, and there are various degrees of confidence. And one can, of course, also look at the non-synonymous variants. But we can see that whole spectrum and examine that, and we'll get data about that. Interestingly, that if we look at databases of drug targets, there's a substantial number of drug targets of interest, including both general targets, ones which already are known to be small molecule targets, and there's some that are already in clinical trials. There's 3 here. So we have homozygous knockouts individuals carry for each of these 3 genes, and as far as I know, they're the first such individuals identified, and would be immediately of interest. There are also knockouts in genes. For instance, these are innate immunity pattern recognition genes, and you might immediately want to go back and ask this individual, is there some class of, have they been subject to certain infection patterns, which would never have gone, would never have gone to a clinical geneticist, but knowing what the gene is allows you to ask questions about their phenotype that might be informative about the function of these genes. So, how can we extrapolate going forwards? Well, in fact, we don't need to extrapolate this. We've got data already, because we've seen in 1,100 people 2,200 copies of each gene sampled from the population. And we can look at how many loss of function variants we've seen in those. In 15,000 people at 6% average IBD, autozygous, we'd get about 1,000 homozygosed copies. So we can sample from the chromosomes we've already observed. And we'd expect to see, I didn't change. These are off because these are for 1,500 people. I edited the wrong version. These are 1.5 times too big. We'd find about 12,000 homozygous rare variants in about 5,500 genes, about 2 per gene in 15,000 individuals. But we're in this space where we hope to access with this phase 1 5,000 genes, knockouts in 5,000 genes, including multiple knockouts on average. Of course, it depends the odds are. So, I think these are things that one really wouldn't have imagined looking at each of these 3 things just a few years ago. In summary, that there are lots of opportunities that are going to be created for increasing our understanding of human genetics by the further increases in the scale of sequencing, which we can really see coming. And we should think about saturation of genetic information in humans. We're going to saturation of haplotypes for imputation and looking at the genomic context. In some sense, that will help us. We'll get some reconstruction of shared ancestry across the world. I think that will we'll place individuals into a picture of shared ancestry. Rather than thinking of sequencing an individual and comparing to one reference, we'll be putting somebody into a pretty complete mesh of the genetic structure of humanity. I think we'll have potential to see all bases mutated, and that will, again, change the way we think about, does this look like the last time we saw that base mutated? What's shared and what isn't? I know that there's context wherever Nico is, and we're not going to cover every possible combination of variants. And we can expect to get information about the effects in humans of homozygous knockout of all genes. And I think that will change the way we think about gene function and genetics in humans. So completeness changes our view on things. So I've covered various people involved. I hope you picked up the names of the people involved in the Haplotech Reference Consortium and in the autozygosity project. I want to thank all my colleagues at the Sanger Institute and collaborators, particularly all the people involved in the 1,000 Genomes project. It's been really, almost as much as the data, it's been a really, for me and I think many others, valuable collection of collaboration that's helped people move forward in their understanding of human variation in sequencing. And also, of course, the ever-increasing number of people who are sequenced who contribute to this. Thank you.
Thanks. I think we can have some discussion. I don't know if this is on. I'm reminded of at the beginning of the 1,000 Genomes project, there was pretty healthy criticism, or rampant criticism at least, of why go and sequence all these unphenotyped samples. So I bet you'll be hearing that again just on the basis of your suggestion here, Richard. And
so it's not all unphenotyped. I think there will be phenotypes attached to these stuff.
Well,
and we have to work out how to use them.
With the, in terms of numbers, the power to have the knockouts is really there, right? It's just not quite organised right to capture that information, and that's probably the most sensitive part of the phenotyping. Is that a fair comment?
So I think something that's been, it's really, we've heard multiple times, is the necessity of sharing information, but the sensitivity of that and the need for doing that appropriately. And there are various models that have been discussed. There's the sort of decipher model and of partial sharing, or we heard from Elizabeth about these strongly secure ways of exchanging data, which is the sufficient data to get at what you want. And I think those have an important place in this era of human genetics research that we're moving into, working out how to not throw away the phenotype information or the detailed genetic information, but how not to compromise people's
terrific talk. So how deeply are these people's phenotype? So we have a knockout, but just to make sense of it, we have to have a deep phenotype also.
So it's not an upfront phenotyping project. And actually, I think that's correct. It's not, I think if you phenotype them all up front, you would never have identified the thing that was actually the relevant thing. So the aim is there will be very, very limited phenotype information captured on entry into the project. And the aim is to be able to go and basically, given a list of genotypes, ask what questions should be addressed in the, or can be addressed across a subset. It's probably a small sets of individuals with particular genotypes. And I think those will, some of them be sort of physiological, and some will be of more clinical relevance. There'll be sets of people who you might think, wow, I would have expected those people to have some particular clinical phenotype. I think it would be appropriate to, but those will each have to go through ethics review.
What about having consent for recontact, so that when you find you have a library of knockouts, that you can basically recontact? I mean, that's
so
kind
Richard,
of what we
is
do.
Richard still here, actually?
Yeah,
or did he have to leave? Richard Trembath. I think he had to leave. The primary care physician will be involved in this consent process. Most recruitment is through primary recruitment will be through your GP in the UK. All follow-up will be through the project. It's not like, you know, this person's name and address is out there and anyone can contact them. They're being consented directly to, and there's been actually quite a lot of outreach already with the communities, in fact with the mosques and the community representatives, you know, independently, and there's quite a lot of support in the community. Since this is not actually it's aimed at healthy individuals, and it's aimed at understanding the genetic health of those individuals and things that are of issue. Sure. Well, that's a standard model for a lot of epidemiological cohorts. Yeah.
Let's go
it's the model.
and then Les, and then we'll quit.
So, Richard, not surprisingly, I follow large sample sets around the world quite a bit. And there are probably about 350,000 individuals well phenotyped that could be sequenced today.
Yeah.
And also, at least in the ERIC study, which I know very well, we spend probably $120 in phenotyping for every dollar in sequencing. And so my question to you is, why don't we, and why haven't we aggressively pursued sequencing those 350,000 samples as one step towards your Million Genomes project, where the phenotypes already exist, and I just really don't understand as a field why we've laid back and we have not aggressively pursued this.
Well, I think there has been an era, and actually I think that's going to continue. I mean, that's exactly what happened with this set of projects like UK 10K. Those are from cohorts which are deeply phenotyped for 20 years, the twins and Alspac cohorts in the UK. Some of the Finnish cohorts we've sequenced 2,000 of those. Sardinia's been sequenced. You know, those people who've been strong proponents of sequencing have been doing that. That's what has got us up to these numbers of 35, and in fact that's not complete, as you know, probably 50,000 or so. I think that's going on. I think that will continue and should continue at some level. But I actually think there are trade-offs. We're sort of entering an era where actually not all of those, well, most of those have been genotyped, not all. There'll be two things in parallel, increasing genotyping and increasing sequencing. That's happening. But we should sort of think, we should think a bit about getting diversity into that sequencing for multiple reasons. Scientifically, in basic science, there are good basic science reasons for ensuring global diversity in the sequencing as we go into the 100,000 range.
I've got Les, and then we'll allow one more.
Yeah.
Okay, that's it. We've got postage.
But I think what's so exciting about what you're proposing, Richard, is this notion of really taking a genomic way of thinking, which is a hypothesis-generating approach to research, and really pushing that into clinical research. And I totally take Eric's point about sequencing phenotype cohorts, and that should happen, and we will learn great things as we already have from doing that. But we have to acknowledge there is a limitation to those phenotypes, which is that they include our presumptions about what the important phenotypes are because they're prospectively phenotyped. What you're proposing to do and what we tried to do with the dominance is put that presumption aside. Let the genomic data tell us what the question should be and then try and answer those in the phenotyping clinical research. And I think what we'll learn is relationships between genotype and phenotype, answers to questions that we don't even know we should be asking today until we see the genomic data that will tell us what we should be looking at.
So alongside genotyping on sequencing phenotype cohorts, I think there's real opportunities in phenotyping sequenced cohorts.
And last.
There was just one last comment about the slide where you're exploring the different spaces from common to rare, and this empty bit in the middle where you only give 2 examples and it's largely empty. But I mean, the whole of the drug metabolism field works in that space and has done for 20 years.
Yeah.
And it's always, this slide is always shown as that space being empty, and that's kind of I think in this room that's not appreciated really. Yeah.
That's a That's good. That's a good point. I think that space is going to fill very quickly in the next, because of the mixture of the sequencing in these cohorts and the imputation into the very large. There are millions of people with genotypes. So I think the combination of that will fill that space and the stuff there.
Well, Richard, I couldn't think of a more perfect talk for this meeting for today. So thank you so much.
Okay, thanks.